Welcome to BioMed Research Digest 2020-60602, today's 12 stories. Story 1, Daily Pill Can Double Survival Time for World's Deadliest Cancer, Trial Shows. Story 2, FDA Approves Oral Wegovy Following Positive Phase III OASIS for Trial Results. Story 3, A Single Protein May Be Holding Back CAR-T Cancer Therapy. Story 4, ASCO 26, Cancer Vaccines and At-Home Injectable Drugs Continue to Show Promise. Story 5, Survival Gains Confirmed. Full Keynote 522 data set to redefine treatment standards in early-stage TNBC at ASCO 2026. Story 6, CRISPR removes extra chromosome in Down syndrome, a breakthrough in genetic research. Story 7, advancing gene-edited stem cell therapy for Friedreich's ataxia. Story 8, adding immunotherapy to radiation improves disease-free survival in localized prostate cancer. Story 9, CRISPR model links faulty collagen to brain microbleeds tied to memory. Story 10, FDA News in Pediatrics, May 2026, Ocrelizumab approved for pediatric MS. Story 11, AG provides patients with better results even after subsequent therapy for advanced triple-negative breast cancer. Story 12, 2026 ASCO Plenary delivers practice-changing progress, Ivonescimab in lung cancer. In a significant step forward for pediatric neurology, the U.S. Food and Drug Administration approved Ocrelizumab, sold under the brand name Ocrevus, for the treatment of relapsing-remitting multiple sclerosis in children aged 10 and older on May 8, 2026. The approval also requires patients to weigh at least 25 kilograms, establishing clear eligibility parameters for this younger population. Why does this matter? Multiple sclerosis in children is a serious, life-altering disease. When it strikes during formative years, it can disrupt education, development, and quality of life in ways that compound over a lifetime. And for decades, pediatric neurologists have had far fewer tools at their disposal compared to their adult-focused colleagues. Most of the powerful disease-modifying therapies were studied and approved exclusively in adults, leaving pediatricians to use medications off-label or settle for a limited menu of options. Ocrelizumab works by depleting B cells, a type of immune cell, through its action on the CD20 protein. This mechanism has been proven highly effective in adult MS populations and is now being extended to children and adolescents with RRMS. The drug is given intravenously, following the same administration route established in adult practice. The implications here are substantial. Pediatric neurologists now have access to a therapy backed by years of adult safety and efficacy data, giving them greater confidence when making treatment decisions for young patients. It also signals that the regulatory environment is becoming more receptive to pediatric indications for complex biologics. That said, important questions remain. Long-term effects of B-cell depletion in a still-developing immune system are not fully understood, and post-marketing surveillance will be critical. Children under the weight or age threshold still lack this treatment option, and the specific clinical trial data supporting this pediatric approval was not detailed in the announcement, leaving the medical community eager for peer-reviewed evidence. Still, this is a genuinely meaningful development, one that may inspire further investment in pediatric neurology trials and ultimately bring more options to a patient population that has long been waiting for them. At the 2026 American Society of Clinical Oncology annual meeting in Chicago, researchers from Dana-Farber Cancer Institute presented landmark data from 2 clinical trials that could fundamentally change how advanced triple-negative breast cancer is treated, potentially saving thousands of lives. Triple-negative breast cancer is among the most aggressive forms of the disease. It accounts for about 15% of all breast cancer cases, but it's particularly deadly in its metastatic form. The 5-year survival rate for patients with advanced triple-negative breast cancer is just 12%. It lacks the hormone receptors and HER2 protein that make other breast cancers targetable by precision therapies, leaving patients heavily reliant on chemotherapy. Sacituzumab govitecan is an antibody-drug conjugate, a smart missile of sorts, that delivers potent chemotherapy directly to cancer cells by targeting a protein called TROP2, which is overexpressed on triple-negative breast cancer cells. It's currently approved as a second-line or later therapy. But the critical question researchers wanted to answer was, what happens if we move it to the very first line of treatment? The answer from 2 large global trials is compelling. In ASCENT-03, which enrolled 558 patients ineligible for immunotherapy, first-line sacituzumab govitecan led to significantly prolonged progression-free survival compared to standard chemotherapy. In ASCENT-04, involving 443 patients with PD-L1-positive tumors, the drug was tested alongside pembrolizumab, an immune checkpoint inhibitor, again beating the chemotherapy plus pembrolizumab combination. Critically, the benefit persisted even after patients went on to receive additional lines of therapy, meaning front-loading this drug doesn't burn bridges for later treatment. And here's why the sequencing question is so urgent. Roughly half of all patients who receive first-line standard therapy never make it to second-line treatment. They never get the chance to benefit from sacituzumab govitecan in its current approved position. Moving it earlier could reach far more patients during the window when they're still strong enough to tolerate and benefit from treatment. If regulatory approval follows, this could be one of the most consequential shifts in triple-negative breast cancer care in years. One of the most talked about results at the 2026 American Society of Clinical Oncology annual meeting came during the prestigious plenary session, a slot reserved for only the most impactful clinical findings across all of oncology. The data, Ivonescimab, a novel bispecific antibody, significantly improved overall survival in previously untreated advanced squamous non-small cell lung cancer. Let's unpack what makes this so significant. Lung cancer is the leading cause of cancer death worldwide. Squamous non-small cell lung cancer makes up roughly a quarter to a third of all lung cancer cases, and it has historically been harder to treat than other subtypes because it has fewer actionable genetic mutations, fewer targets for precision medicines. The arrival of PD-1 and PD-L1 checkpoint inhibitors was a major step forward, but survival gains have begun to plateau. Ivonescimab takes a different approach. Rather than blocking just one pathway, it's a bispecific antibody, a single molecule engineered to hit two targets at once. It simultaneously blocks PD-1, the immune checkpoint that cancer uses to hide from the immune system, and VEGF, the protein that tumors use to grow new blood vessels and, importantly, to create an immunosuppressive environment around themselves. By cutting off both escape routes at once, Ivonescimab may produce synergistic effects that neither target alone can achieve. The trial compared Ivonescimab plus chemotherapy not against a chemotherapy-only control, but against tislelizumab, an already approved and active PD-1 inhibitor plus chemotherapy. Beating an established immunotherapy regimen in a head-to-head survival comparison is a genuinely high bar, and Ivonescimab cleared it. The implications for global treatment guidelines are immediate. Oncologists, guideline committees, and regulators around the world will be scrutinizing the full peer-reviewed data. If it holds up, and the plenary selection suggests the oncology community believes it will, Ivonescimab could become the new first-line standard of care for squamous non-small cell lung cancer and potentially open the door to a broader era of bispecific immunotherapy across solid tumors. At the 2026 ASCO annual meeting, personalized mRNA cancer vaccines, including intismeran autogene combined with pembrolizumab KYTRUDA from Merck and Moderna, showed a 59% reduction in risk of distant metastasis and an overall survival rate of 92.2%. These results reinforce the transformative potential of mRNA-based cancer immunotherapy. The data is expected to shape treatment standards for multiple cancer types. The final analysis of the Phase 3 KEYNOTE-522 trial confirms sustained survival gains with pembrolizumab-based combination therapy in early-stage triple-negative breast cancer (TNBC), a historically aggressive and difficult-to-treat subtype. These results are expected to redefine standard of care for early TNBC. The data was presented ahead of the 2026 ASCO annual meeting. Researchers have used CRISPR gene editing technology to successfully remove the extra chromosome 21 responsible for Down syndrome in laboratory models, marking a conceptual breakthrough in chromosomal disorder research. While not yet a clinical therapy, this proof of concept opens new avenues for understanding and potentially treating trisomy conditions. The achievement demonstrates the expanding scope of CRISPR beyond single gene editing. Researcher Stephanie Cherqui discusses the development of a CRISPR/Cas9 edited hematopoietic stem cell (HSC) therapy for Friedreich's ataxia, a progressive neurological disease with no cure. The approach involves editing patients' own stem cells and reinfusing them to halt neurodegeneration. The work is advancing toward clinical translation. A Johns Hopkins study found that adding the investigational immunotherapy aglatimagene to standard radiation therapy improved disease-free survival in men with intermediate or high-risk localized prostate cancer. This combination approach could offer a meaningful advance for a patient population that currently has limited immunotherapy options. The findings were released in conjunction with the 2026 ASCO meeting. Using a CRISPR-based disease model, researchers discovered that genetic variants in TIMP2, which regulates the enzyme responsible for breaking down collagen IV, are significantly associated with brain microbleeds linked to memory and cognitive decline. This provides new mechanistic insight into cerebral small vessel disease and potential targets for intervention. The findings have implications for understanding vascular contributions to dementia. On May 8, 2026, the FDA approved intravenous Ocrelizumab Ocrevus for the treatment of relapsing remitting multiple sclerosis in pediatric patients aged 10 and older weighing at least 25 kg. This marks a significant expansion of MS treatment options for younger patients, where treatment choices have historically been limited. Ocrelizumab is a B-cell depleting anti-CD20 monoclonal antibody with established efficacy in adults. Dana-Farber researchers report that sacituzumab govitecan, an antibody-drug conjugate ADC, provides superior outcomes for patients with advanced triple-negative breast cancer even when assessed after subsequent lines of therapy, strengthening the case for its use as a first-line treatment. The findings support regulatory approval of the ADC in this aggressive breast cancer subtype. TNBC has historically had very poor prognosis with few targeted treatment options. At the 2026 ASCO Plenary session, Ivonescimab, a novel bispecific antibody targeting both PD-1 and VEGF, combined with chemotherapy significantly improved overall survival compared to tislelizumab plus chemotherapy in previously untreated advanced squamous non-small cell lung cancer. The results signal a new era of bispecific immunotherapy in thoracic oncology. These findings are expected to influence global treatment guidelines.